Let's now consider the mechanisms in place to repair more extensive physical damage to the DNA, resulting in breaking the DNA strands. The first of these mechanisms we will consider is referred to as non-homologous end joining. As shown at the top of our figure, there has been mechanical damage to the DNA resulting in a double-strand break, or DSB. Note that cell division cannot occur until the DNA is replicated, and this presents a big threat to that process. The cell therefore employs a repair mechanism that is error-prone in order to prevent a result that would be lethal to the cell. To understand this process, recall that homologous DNA contains like sequences. In this mechanism, Q protein, pictured as the red dual circles, binds to the broken ends of the DNA and then recruits repair proteins. The space is filled in with nucleotides, but since there is no template to follow, misincorporations are all but inevitable. Notice that the ends are joined in a non-homologous manner. In other words, the sequences don't match. Another way to repair a double-strand break is through genetic recombination, a process of rearranging the genetic information to form new associations. In homologous recombination, this process involves a reaction between homologous or like sequences. In contrast, non-homologous recombination is the combination of different nucleotide sequences. These recombination events occur in specific zones within the chromosome and are called hotspots. The physical exchange of parts of the chromosome was proven in an experiment conducted by Messelson and Weigel. Two types of bacteriophage were used. These are viruses that infect bacteria and do so in a very specific way. The same bacterial host was infected with two different phage particles, one carrying a heavier DNA molecule, pictured as the blue phage, and another carrying a lighter DNA molecule, the pink phage. The virus was allowed to complete its life cycle, resulting in lysis of the host and release of new phage particles. Results showed that although there were phage particles containing solely the heavy or the light DNA, there were also particles carrying a recombinant mixture, shown below as the purple phage particles. Having proven that recombination is a naturally occurring process, let's examine in our next lesson the steps in that process.